And then you wait until it cools, and now you have a candle again, right? So the way to get the color into the candle is to get it hot because it's wax. So these cells have almost like that wax candle layer around them, right? You ever drip wax on your clothes when you're doing this? You'll stain them, right? You can't get it out. You have to actually iron out. That's how you get a wax stain out of your clothes. You put paper, I know this from experience, right? You put a paper bag underneath and you iron on top of it and it melts the wax and it soaks into the paper towel because you have to get it hot, right? If wax is cold, it's impenetrable. If a mycolic acid layer is at body temperature, it's impenetrable. Which means that you can disinfect, right? You can spray Cavicide on these bacteria. You can give antibiotics to it. You can do all kinds of things, and if it can't penetrate the cell, it can't kill the cell. So something like tuberculosis, which is caused by one of these bacteria, is highly resistant to disinfection. It's highly resistant to antibiotics, which makes it a real problem because it also is highly contagious, right? All you do is cough it on somebody or cough it on the countertop, and you can clean that countertop with Cavicide all you want and it will not die, which is why it easily spreads through a population, right? So these cells that have this outer membrane filled with mycolic acid are called acid-fast bacteria. And you're like, acid-fast, right? So notice mycolic acid is an acid, right? So they had no idea really what was inside these cells. What they did was they would put a stain on them and then they would pour acid to see if the acid could remove the stain. And from most cells, the acid will remove the stain, but from these, they would hold fast to the acid, right? They would hold onto it, so they called them acid-fast, meaning you spray acid on them and the stain doesn't come out. Because once you get the stain in, you can't get it back out, right? Just like once you take your candle and you dip it in colored wax and you dry it, you're not gonna get the color out of your candle, right? If the color goes in when it's hot, then it stays. So we actually are gonna do this technique, and what we do is we heat up the cells, so we melt the wax and then we stain them while they're warm to see if we can pull it back out. And when we can't, we call them acid-fast. When we can, we call them non-acid-fast. So if you've ever been in a clinical setting, you might have heard somebody say, we're ruling out AFB, or we're doing an AFB test. What they are typically looking for is tuberculosis. They want to know whether a patient who has a respiratory illness has tuberculosis, and so they order what we call an AFB test. If it comes back AFB positive and it came from your lungs, it's either leprosy, which can't grow in your lungs, or it's tuberculosis, right, most of the time. So that AFB test is one you'll hear if you're ever in clinical settings. If you hear that, that means they think that person has tuberculosis, because Mycobacterium tuberculosis has that waxy mycolic acid layer, right? And that means that they have to give special antibiotics that are able to penetrate that mycolic acid layer, and they have to use special disinfectants, which are capable of killing those bacteria, right? Because you got to get that disinfectant through that mycolic acid layer, through the peptidoglycan into the cell to damage the cell, right? So these tend to be very hardy cells. So if we kind of make a continuum of, right, cells, and we're going to talk a lot more about this when we get to chapter 7. But you and I, we are the biggest wussies. It's easy to kill ourselves, super easy. Next biggest wussy would be a gram-positive cell. Next biggest wussy, who really isn't a wussy at all, would be a gram-negative cell, and then beneath him we would have those acid-fast bacteria. And you can imagine, right, because they have more layers that you have to get through, right? These guys have a really thick peptidoglycan. They have a thin one, but they have that layer. These guys have a really thick peptidoglycan and that mycolic acid outside of them, right? And you got to go from outside to inside to kill them, and it's harder to do that, right? So these cells, we actually do a different kind of staining technique for. We call it acid-fast staining. You're not going to do that one on Thursday. You're going to do that one next week. Acid-fast staining uses heat to melt the mycolic acid, to melt that wax, and then stain so that we can get through it and color them. And if we can, we have an acid-fast bacteria. Now I just said tuberculosis is acid-fast, and then I said you're going to be practicing this technique. You're not going to be doing it with tuberculosis. You're not going to be doing it with leprosy either, even though leprosy is not very contagious. Right? If you read the Old Testament or you watch Ben-Hur, you have this idea that leprosy is this really terrible thing, right? You will find them in the valley of the leper, right? We take lepers and we get rid of them, right? Well, sadly enough, we still do that today, but you know how you get rid of leprosy? Antibiotics. And it's really not that contagious. You can live alongside a person with leprosy, but we don't use that either. We use an organism named Mycobacterium smegmatis, which is actually part of the normal flora. The urogenital tract, we did not take it from a person, we grew it in a lab. Much like we did not take our E. coli from someone's stool, we grew it in a lab. But that, because it's a member of that genus, Mycobacterium, will also be acid-fast. So it allows you to practice that technique with a safe organism, right? A biosafety level 1 organism, not tuberculosis, which we would have to have respiratory gear to deal with, right? Hands goes over here.
On this slide, how we're going to distinguish this is the acid-fast or gram-negative?
So what you have to be careful of, right? So you guys have, well, I don't have to get another, you have this in your drawer, right? So you have 1, 2, 3, 4, 5 different stains, all of them stain bacteria. Sometimes we apply heat, sometimes we don't, right? But all of these will color bacteria because they all have a positive charge and every cell has a negative charge. So the thing about staining is you have to know what the technique is, not just the color. So for a gram stain, we use these stains right here. We use this purple one, and then we decolorize, and then we make it pink. So the first color is purple. So gram positive or purple, gram negative or pink, which is the second color. For the acid fast, you are going to use, these are darker, right? So it's harder to see, but you are going to be using something called carbol-fuchsin, which is a fuchsia color, it's bright pink. And then you're gonna decolorize, and then you're gonna use methylene blue. And guess what color that is? Blue. The first color colors the acid-fast bacteria. So in this case, the positive is pink, and the negative is blue. So you have to be careful. You don't want to think in your mind, purple is gram positive, pink is gram negative. If you want to make that kind of a connection in your brain, think, the first thing I use is positive, and the last thing I use is negative. So the color of the first thing you use is the color of the positive. The color of the last thing you use is the negative. So if I go back, right, because if I look at that picture, it looks kind of similar to that picture color-wise, right? But if I tell you the first color that I used here was purple, the second was pink, is it the bacilli or the cocci that are positive if the first color is purple? The cocci are positive. If I tell you the first color I used was fuchsia, or bright pink here, and the second color I used was blue, then which of these two is positive? The bacilli. I know you want to say rod, the bacilli. Now, notice something else about these bacilli, and then I'll get to you. I haven't forgotten that you had your hand up. Notice here, they're all kind of by themselves, right? The bacilli are all kind of just hanging out. They look kind of clumpy and stuck together here because they're waxy and they stick to one another. So when we see that, we go, oh, that's interesting. I see a bacillus that's gram positive, but it looks a little weird when I gram stain it and they're all stuck together, and I see to myself, I should really do an acid-fast stain on this because that could be acid-fast bacteria because it's gram positive, it's bacilli, and it looks sticky. And now, what was your question?
For the on the board, for the how is it easiest to kill? What do you mean by we, like our cells?
Like our cells. You and I. You're the easiest, right? Which makes disinfection and antisepsis tricky, right? Because what's the easiest thing to kill on your body? You are. Right? So we have to be really careful. Most of the things that we say are not safe for humans, right, and we can't use for antisepsis, like cavicide. Right? If you spill bacteria on yourself, you do not spray cavicide on yourself. Because that's not safe for your cells. It will kill bacteria, it will also damage your cells, right? Like bleach. Bleach is a great disinfectant. You swallow bleach, you're gonna have a bigger problem than the bacteria that are growing in your gut, right? So we are the biggest wusses, and then they follow us. And really there's a big difference, right? There's a big space here. We're way wussier than bacteria are, right? Like your cells, for instance, have to, for the most part, be bathed in a solution that has a pH of 7.4. And if it goes to 7.45, you freak out. If it goes to 7.35, you freak out. E. coli can be in an environment that goes from pH like 4.5 to 9, and it hardly even phases them. Their internal pH will change by 0.1, right? So they'll have 1,000-time change in the pH outside, and that will amount to a 0.1 change inside. So they're really, really way more tough than we are. All right, so that's our acid-fast bacteria, right? So cell wall prevents osmotic lysis, that's like everybody else. Then we have the peptidoglycan, which we know ranges from big and thick to really thin. If it's thin, we have a lipopolysaccharide outer membrane, and then we have this other exception where we have thick peptidoglycan, so we have gram-positive, but they also have this mycolic acid layer. I keep losing my color, there's my clicker. All right, so now, finally, are you ready? We're in. Plasma membrane. We finally, right after how long, we're in the cell. Now here's the good news about the plasma membrane. It's just like yours. Lipid, bilayer, semi-permeable, right? All of those things that you learn in general biology about a cell membrane, a plasma membrane, are the same here for all bacteria. You go, whew, that's easy, right? But there are always exceptions. And this is the thing about being a person who studies tiny things, right? Things that look the same are not really the same, right? So like all those prokaryotes, they all look the same. And then we use the gram stain technique. We're like, oh, it turns out they're different. And then we add acid fast technique. Oh, it looks like they're different again. And then you have all these different things. And then they started looking at all of those bacteria, right? And all the guys that we put down here, the gram negatives, which would include gram negative bacteria and gram negative archaea. Ooh, I didn't put my phone on silent. That's my daughter. See, now whoever it was that family member dinged them with the fine. I do that to my kid all the time. When they don't answer their phone, I do a find my phone alert on their phone. Now you don't have to feel bad because mine did the same thing. All right. So, gram negatives, little to no peptidoglycan, not enough to hold on to that, right? So, we have all of those in a group, and people are probably like, oh, finally, we've got them all separated into groups, right? Because what else could be different? And it turns out that there are a whole group of organisms who have a plasma membrane like nobody else on the planet. So you, fungi, plants, protozoans, everything that has cells made with a nucleus inside them, we call them eukaryotes. All of you and all of the bacteria have a plasma membrane that looks like this. Those little guys who don't have any peptidoglycan but seem gram negative, some of them don't have this kind of plasma membrane. And it turns out they're the same guys that have that pseudomerian. They have a totally unique plasma membrane, right? So in their DNA is a recipe for a plasma membrane that is not the same as anything else on the planet, which makes them fundamentally completely different. Even though we took everybody without a nucleus and said, you get to be in Kingdom Monera because we don't really care about you. You all look the same, you're boring, we'll stick you all together. Well, it turns out when we started looking at them, right, we found the gram thingy, we found the acid fast thingy, and now we found that there are organisms who have a totally different plasma membrane. So how different is it? It, in a lot of cases, is a monolayer. So rather than being two separate phospholipids, right, who orient themselves, right, so here's the hydrophilic head and the hydrophobic tail, and here's the hydrophobic tail and the hydrophilic, right, so they sort of orient themselves like that. They have one single phospholipid that has a hydrophilic head on one side and a hydrophilic head on the other side. So that's one way. The other way they're totally different, and I know it doesn't seem like much, right? It's actually just one single oxygen atom, but the way that they put theirs together are totally different from every other organism on the planet. Every other organism on the planet, right? So plants, fungi, protozoans, protists, animals, which includes you and me, all the bacteria, we do this. We have a little oxygen there. We call that an ester linkage. If you've taken organic chemistry, right, you remember esters and ethers. Who cares, right? I'm not gonna ask you to draw it. What I want you to do is think about how fundamentally different a cell is that does not do that, but instead has this kind of linkage here. So fundamentally, these cells that we threw together into Kingdom Monera, and this is something we're going to talk a lot about when we get to our diversity unit. These cells that were thrown together because they had no nucleus. So what could there possibly be different about them? Because they all don't have a nucleus, and all these other guys have nuclei. Look at how different they are, right? So we'll throw them all in a group. Then we found out their cell walls are totally different. And now we find out that the cell membrane of archaeans, a subset of the prokaryotes, has a totally different plasma membrane. Now, I'm not going to ask you to draw this. I'm not going to ask you who has an ester or who has an ether. What I want you to do just right now, because remember this unit, a lot of it is just laying the groundwork in these early chapters. And what we're doing right now is laying the groundwork for the diversity of microorganisms. The kind of cell wall, the amount of cell wall, the things that are accessories to the cell wall, like a lipopolysaccharide outer membrane or a mycolic acid layer, and the fact that they have different plasma membranes. These are things that add to the diversity of what until very recently were all thought to be just a bunch of little dudes who don't have nuclei. So that's what we're doing right now, right? We're just setting the stage for diversity. Now, you might think, plasma membrane, right? You're like, sweet, plasma membrane, not much in there other than that. What could we possibly talk about? Just wait. Remember we said, just because they don't have nuclei doesn't mean there's nothing going on, right? These are incredibly complex cells that are capable of doing all sorts of really interesting things, eating interesting things, doing interesting things, right? So just because we got inside the cell doesn't mean the story's over. Because, right, we say that bacteria and prokaryotes, right, are cells who lack a nucleus, but technically what we should say is they lack membrane-bound organelles. A nucleus is a membrane-bound organelle. So is an endoplasmic reticulum, so is a Golgi body, right? So most of those organelles are used to compartmentalize work, right? The nucleus compartmentalizes the recipe book. The Golgi body compartmentalizes protein sorting. The endoplasmic reticulum compartmentalizes protein synthesis. The lysosome compartmentalizes the digestion of unwanted stuff, right? So they're just compartments in the cell, but they're membrane bound. Prokaryotes, because they still have to compartmentalize their jobs, do have organelles. They're just not membrane bound. So in order to not confuse you, they don't call them organelles in prokaryotes. Rather than calling them organelles, they call them inclusions. But an inclusion, you can think in your brain, if it doesn't confuse you, this is like an organelle. It's a compartment in the cell where a particular job is done. Or where a particular something is stored, right? Like a nucleus is a membrane organelle where the DNA is stored. A metachromatic granule, which is a mouthful, is an organelle where phosphate is stored. Now, everybody can imagine why DNA is compartmentalized, right? Because it's your recipe book, right? It's your precious recipe book that was passed down to you through the generations, right? So you want to take care of it, right? You never let it leave the nucleus. But why would you want to hold on to phosphate? Well, let's say that you wanted to replicate that piece of DNA. You need A's and T's and C's and G's. You need ATPs and CTPs and TTPs and GTPs. And what's the P at the end of ATP and GTP? It's phosphate. So why do you need phosphate? Because you want to make DNA, you want to make RNA, and you want to make phospholipids. So if you want to make any of those things, you have to eat phosphate. You have no problem, right? You can go to the grocery store and you can buy food and you can have whatever you want to be nutritionally complete. These are bacteria that are wandering around in a harsh environment, and so whenever they come across something that they can use, they store it. And whenever they come across phosphate, they store it. And they store it in these things called metachromatic granules, right? So a granule, the reason it was called a metachromatic granule is because when they stained these cells with a stain, notice those little areas that are clear and then they have a dark center inside them? They said, ooh, that looks like a metachromatic granule. It's a granule, it's a thing, it's a little piece that gets colored, metachromatic, right? So it's a colored particle. They didn't know why it was colored. They just found it, and they were like, huh, I wonder what's inside that. Turns out it's a bunch of phosphate. So lots of bacteria have metachromatic granules that they use to store phosphate, because no phosphate, no DNA replication. No DNA replication, no cell division, you're dead, right? Because what's the meaning of life? To make babies of yourself, right? That's what bacteria live for. And if you can't make new DNA, you can't make a baby cell, and you're out, right? You're gone, and somebody else replaces you in the next generation, right? So survival of the fittest says you gotta have phosphate, so when they find phosphate, they grab it and they store it so that they can use it. The next, whoops, I went backwards instead of forwards, right? The next three are all energy reserves, right? So you have energy reserves. Like bacteria, you store carbohydrate. The carbohydrate you store is glycogen, right? If you've taken physiology, you got to learn all that fun stuff about glycogen breaking down into glucose, right? Your liver does this job for you. Carbohydrates are a great energy source because they're really easy to break into sugar, right? Which is what glycolysis needs to work, right? So what's your main energy source regardless of what you eat? You could be completely ketogenic. The carbohydrate never passes your lips, and you have a blood sugar that is constantly maintained by a liver that is converting other things into sugar and sending them into your body. Most importantly, because the only thing your brain can eat is sugar, right? So you store, but what your body would like is carbohydrates, right? Now we have this problem where we eat too many carbohydrates, right? So we have to think about a time when maybe you're like, it's the 1800s and you're in a covered wagon, you're cruising across the country, right? You don't care what kind of thing you're eating. If it gives you energy, you eat it, right? Carbohydrates are a great source of energy because all you have to do is cut them into little pieces, and those little pieces are sugars and they go right into glycolysis. So carbohydrates would include glycogen, like people use, and polysaccharides, which are what bacteria store. Those polysaccharides, just like the carbohydrates that you store, are a nice energy reserve for them, right? So when life is good and they've got plenty to eat, they make ATP, now they have a surplus of ATP, so they convert it into other molecules and ultimately into polysaccharides so they can store them. Just like you do, right? You eat lunch before you come to class, you do not need all of the energy from your lunch right now. So your liver does the job of taking and storing all of those materials, right? Same thing they're doing here. So polysaccharide granule is their carbohydrate storage, and it's pictured here, these big giant things. So you can see how much of the cell body can be devoted to storage. Because they're on a covered wagon ride their whole life. They never know where their next meal is coming from. So they store it, right? If they can, they store it. Now, what if they don't use that energy, right? Because as you can see here, polysaccharides take up a lot of space. So what do? If you've eaten a big lunch, you come to class, you sit for 5.5 hours, and then you're tired, and so you watch TV and you go to bed. What does your body do with the glycogen that you stored? Turns it into fat, right? Because fat, you can pack much more tightly together, right? And they do the same thing. For long-term storage, they take those polysaccharides and they convert them into fats, right? So we call those lipid inclusions, and they're pictured here. So notice, much smaller fat can be much more tightly packed in them, which leaves room for all the other stuff. Now here's a weird one. Also, under the energy reserve is sulfur. You're like, okay, I can picture the carbohydrate, I can picture the fat. Why on earth are you saying that sulfur is an energy source? I'm saying that because there's a whole group of bacteria and archaea that actually eat sulfur. Weird, right? And you don't eat sulfur, not at all. You do need some sulfur, right? Certain proteins, certain amino acids have a sulfide residue in them, like cysteine. So you need a little bit of sulfur, but that's not what this is for. This is not to make amino acids. This is actually because they eat sulfur, right? They eat sulfur. So these are a bunch of sulfur granules lit up inside of bacteria. So they actually will take in sulfur. And these guys are super cool. We're going to talk about them in chapter 5 because they can actually be the primary producers for a community, right? Like a plant is normally a primary producer, right? It makes food and then something eats it and then something eats it and something eats it and all the way up. In the bottom of the ocean, we have deep sea hydrothermal vents which spew hot sulfur into the ocean, and these guys are down there eating it. And then something eats them, and something eats that, and something eats that, and they actually are the basis of that food web down at the bottom of the ocean because they eat sulfur. So if you eat sulfur, then it makes sense, right? If you happen to be cruising through a sulfide-rich environment like the Salton Sea or Mono Lake, you're like, ooh, sulfur, and you grab it and you store it, right? Because you're gonna need it later on, and you don't know when you're gonna find it, so you hold on to it. So those are all energy reserves, right? So like a mitochondrion taps energy from different things, right? You can have these as taps for things, or like a chloroplast, right? Taps different organelles to make energy. You can have a bacterium or an archaean who uses organelles, they're just not membrane bound, right? So that's the distinction, is they're not membrane bound. So speaking of plants and those sulfur oxidizing bacteria in deep sea hydrothermal vents, which we will talk about at the back side of chapter 5, are all what we will call autotrophs. Autotroph means self-feeder. They make their own food, right? So photosynthesis is a kind of autotrophy. Photosynthesis, you'll remember from general biology, is divided into two things, right? So here's where you can test whether you need to go review. Photosynthesis is divided into the light-dependent portion and light-independent portion. The light, right? The light drives the production of ATP, and it produces something called NADPH in plants. And then those light-dependent reactions, which produce ATP and NADPH, go to the light-independent portion, which is where CO2 is built into a sugar. Right? That's photosynthesis in a nutshell. That second portion, the light-independent part of photosynthesis, is called CO2 fixation or carbon fixation. Because whenever we take something inorganic and we convert it to its organic form, right? So commonly we'll say that CO2 is turned into a carbohydrate, right? And that's abbreviated CH2O, right? So the difference between them is this guy right here. This is inorganic, this is organic. Whenever something inorganic is converted to its organic form, you will oftentimes hear that called fixing. We have carbon fixing, we have nitrogen fixing, we have sulfur fixing, right? You probably didn't call it carbon fixation when you were in general biology because you had enough to do, right, with your instructor, just trying to wrap your mind around the idea that somehow sunlight gets converted to ATP, right? Light energy gets converted to chemical energy, and somehow that chemical energy is used to build CO2 into a carbohydrate. But we call that carbon fixation. That occurs inside the chloroplasts in plants, right? So they compartmentalize that job. That job in bacteria can occur in what's called a carboxysome. So a carboxysome is a place where carbon fixation can occur in bacteria, much like the chloroplast of plants. That job can be compartmentalized to a particular place. We're almost done. We got two more. So hang tight. The last two, I would say, they're kind of my faves of all of the inclusions, right? So they're fun. The second to the last is something called a gas vacuole. Now, incidentally, this is not an anatomy class. I am not ever going to show you a picture of something and say, what is that thing? So you don't have to like go memorize what all these pictures look like. I'm not going to show you this and go, what is that thing pointing to? It's just to show you, right, what a gas vacuole looks like. It looks like each of those little clear circles inside that cell, and it is filled with, guess what? Gas. Gas is light, right? It's not heavy. I mean light like this light. I mean like it doesn't weigh a lot, right? So gas allows you to float, right? Like if you don't swim well, you can put a vest on, right? And that vest can just be filled with air, and that will allow you to float at the surface of the water and make you buoyant. Buoyant is not a science word. Buoyant is a word that means something floats, right? It has less density than what it's in, right? So air, right, you can try this at home. It's awfully hot today. It'll probably still be warm in your pool. If you have one when you get home, you go jump in the pool and lie on your back and inhale and hold your breath and feel yourself rise in the water. And then exhale and feel yourself fall, right? Because air changes your buoyancy. When you have more air, you're more buoyant. So let's say that you are even though you're motile, you are a bacterium that lives in the ocean, right? Which is deep, thousands of meters deep, but you need to be in the top because you photosynthesize. So you could spend your whole life swimming to try to stay at the top of the ocean, right? Just like a person, if you fall off of a ship in the middle of the ocean, you could swim for the rest of your life and try to stay at the surface of the ocean, but how long is your life going to be? Because you're burning all of your energy trying to swim at the top of the ocean. Not very long, right? You're gonna drown. Because ultimately you can't keep burning energy to stay afloat. But if you had a life vest, you wouldn't have to expend any energy staying afloat in the water. And that's what these guys do. They have these gas vacuoles, and it's like wearing a life vest. And it allows them to sit right in the top of the water so that they can always photosynthesize and they don't have to use any energy to do that. So they sit at the top of the water and they hang out and they photosynthesize, right? So they always stay where it's lit. There are actually other bacteria that can go to the top and then come back down. Like when it's dark out, maybe they want to go and like catch some of the fertilizer that's in the ocean, they'll come down out of the photic zone and they'll collect stuff, and then tomorrow morning when it's light again, they'll increase that gas and they'll go back up again. So they'll actually use that to move, and it's a more passive process, which means it takes far less energy. And what we're going to learn in chapter 5 is it's all about stockpiling energy, because energy is what leads to growth, and growth means that you're around for the next generation, or at least your DNA is, right? Which is the meaning of life in terms of biology. We could get all existential about it, right? But what's the meaning of life? To pass your DNA on to the next generation so that your DNA is still around. And you need energy to do that. So chapter 5, we're going to learn all about all the ways cells make energy. In chapter 6, we're going to learn how they use that energy to make baby cells. So all of the things that they can do passively mean more energy can be devoted to making babies, which is their main goal in life. All right, last one. This is my all-time favorite. It's called a magnetosome. It's a magnetic body. Some, lysosome, peroxisome, carboxysome, magnetosome. Some means body, right? Like you have somatic cells in your body, which includes everything except for your eggs and sperm, right, which are germ cells. Somatic just means body. So a peroxisome is a body filled with peroxides. A lysosome is a body filled with lytic enzymes. Carboxysome is a body in which CO2, which is carb, right? That's the carboxy from that. Magnetosome is a magnetic body. Here's why I like it. First of all, it's very tidy. I'm a very tidy person. I like tidy things. Like on my way to work today, I went to the Container Store, which is one of my favorite things to do because I love tidying things up. And my son started homeschool this year, so I have this list of stuff that I wanted, right? And look how tidy this is. Look at it, they're all lined up perfectly. Number 2 reason I love magnetosomes is if you had them on your microscope slide, you could have a magnet underneath and you could drag them all over the place because they're magnetic. Now, they do not have that because they're tidy, and they don't have it because I derive joy from dragging them around a microscope slide. They have magnetosomes for what may seem like the strangest reason on the planet, and that reason is the iron oxide, which is what makes that magnetic, destroys hydrogen peroxide. You're like, oh, I get it. Hydrogen peroxide, which I would put on my cut. So if this guy landed on my body and I had a cut and I put hydrogen peroxide on him, he would die, except for he has this magnetosome, so he can survive hydrogen peroxide. And you would be 100% wrong. That is not why he has that. He has this because wherever there is air, there is hydrogen peroxide, which seems weird, right? Hydrogen peroxide is a byproduct of all aerobic metabolism. So right now in your body, you are creating hydrogen peroxide, and it's a poison. Hydrogen peroxide is a nasty reactive molecule that we're going to talk about a lot in chapter 6, that sort of like, you ever watch the old cartoons with the Tasmanian devil, right? The Tasmanian devil spins and just bangs into things and destroys everything he comes into contact with. That's hydrogen peroxide. So imagine hydrogen peroxide in your cell, just spinning like a Tasmanian devil, right? Banging into DNA, banging into ribosomes, banging into the cell membrane, banging into everything, destroying everything. That would happen in every cell that lives in an aerobic environment, including mine and yours and his. So he has iron oxide so that he can destroy that hydrogen peroxide and protect himself. You have an enzyme called catalase, right? What happens if you have a cut, you put hydrogen peroxide on it? It bubbles. You know why? Because you have an enzyme called catalase that takes that hydrogen peroxide and breaks it into water and oxygen gas. That's your protection. You make catalase to protect you. He has these little bodies filled with iron oxide to protect him. So every organism who is going to live where there is oxygen has to protect itself from oxygen, because oxygen is a very reactive element. Not O2, but all of the things that O2 will very easily become, right? You get more and more concerned about this as you age, because they can make you get wrinkles and look old because they're free radicals, right? You've ever heard, oh, you gotta eat grape seeds because grape seeds are high in antioxidants, right? You gotta put lotion on your face because the free radicals in the air will damage your skin and make you get wrinkles. Those are all true. Those things won't really work because what you need is something inside the cell stopping that damage from happening. So we're going to talk a lot about those free radicals, those reactive oxygen species in chapter 6, but for right now we're going to say one of the ways that you can avoid damage even though you live in an aerobic environment is by having a magnetosome. You don't have them, I don't have them, lots of bacteria don't have them, but some bacteria do, and the reason they have those magnetosomes is to enable them to live where there's oxygen present. Because the other thing about oxygen is that you absolutely need it, right? I mean, hold your breath long enough, what'll happen? You'll die, right? That vital force that we were like, oh, dummies, what, you know, vital force, it's oxygen, right? You need oxygen. We're gonna learn on Thursday, it sits at the bottom of your electron transport chain and collects electrons for you, and if you don't have it, you die. Because you don't make ATP, you can't contract your muscles, you can't send nervous impulses, and you're dead, right? So everybody needs oxygen, almost. And if you need it, you have to have a way of dealing with it when it gets crazy, right? Because oxygen's like a toddler, right? It's just 1 second away from going, it's like a toddler who went to Disneyland and it's 11 o'clock at night, right? They're right there, right? Anything can send them over the edge. Oxygen is a very temperamental molecule, so you have to have a way of protecting yourself, which we're gonna talk a lot about in chapter 6, but for right now, that is what a magnetosome is for. All right, which leads us to the very interior of the cell where we have DNA. And yay, DNA here, exactly the same as your DNA. Double-stranded, semi-conserved, they replicate, right? All of that stuff is the same, so all we have to do is say DNA, right, which is the hereditary material of the cell. One interesting thing that we're going to talk a lot more about in chapter 8 is that though they have a single piece of DNA, they also have this extra chromosomal DNA called plasmids. And so in chapter 8, we're going to talk a lot about the uniqueness of the prokaryotic chromosome and also the plasmids that they contain that allow them to have new traits, right? Because DNA, of course, codes for the messenger RNA, which gets translated into proteins. So if you got the gene, you get the protein, right? So if you have new DNA, new genes, new proteins, new traits, like, for instance, antibiotic resistance, right? We have bacteria that didn't used to know how to eat antibiotics and dismember them, and now they do because they got new DNA and it changed them, right? All right, so we have this here, right? So we're the wussies, then the gram-positives, then the gram-negatives, then the acid-fast, and at the very bottom, we have what are called spore-forming bacteria. Now, spore, there's a big distance between those two. Spores are not like fungal spores, right? So in general biology, I know that there's a lot where you look at fungi, right? And you look at them like a bread mold under the microscope and you'll see these spores and they're reproductive. These are not reproductive. Bacterial spores, right? If I say that the ability to form a spore makes you super resistant, right? Keep that in mind, right? That doesn't mean you're reproductive. Resistant usually means, right, we are both best protected when we hunker down, right? You're best protected when you hunker down. So there are certain bacteria who, among all of the other genes on their chromosome, contain about 100 genes that are completely devoted to forming spores. Now, E. coli is not a spore former. So as your bacteria have grown, they start to run out of space, and they start to run out of food, and they start to build up waste products around them, right? So they're all like peeing in the same pool, right? So life is not good anymore inside of those test tubes, and all they can do is the same thing you would do, which is die. But there are certain bacteria who have the genetic information when things are bad to hunker down, right? Kind of go into the bacterial equivalent of a fallout shelter. And what they do, right, is they go through this process, and it takes a bit of time. It takes about 8 to 10 hours from the point where they say, I think I should form a spore, to the place where they're actually forming a spore. So they're still vulnerable, right? But it's this lengthy process basically where you take a new piece of DNA, right? Your new shiny pristine piece of DNA, and you just pack it and pack it and pack it and pack it and pack it. Like, have you ever been moved by a professional moving company? So, I mean, I'm not fancy, but we're in the Navy, right? So when the Navy moves you, really the only good thing about being told, in 2 months you're gonna move to the other side of the country is, oh, but don't worry, we'll have people come and pack your stuff up for you. It's real fun until you're unpacking it, right? So you get this giant box, and you open the giant box, and you see this big, huge wad of newspaper. And you start rolling the newspaper apart, and you roll it, and then there's another piece of newspaper, another piece of newspaper, another piece of newspaper, and finally you get to the inside of this huge thing, and there's a single glass. You're like, thanks. You probably spent more on newspaper than I spent buying that glass because, you know, like I'm cheap, right? So, but that's kind of like a spore because the DNA is way more precious than a glass. So you take the DNA and you wrap it, and then you wrap it again, and you wrap it again, and you wrap it again, and you have these thick layers of peptidoglycan, and then you put a spore coat, and now it becomes so impenetrable. Do you know they have found spores, right? So this spore, once it has been formed, they have found them in Egyptian sarcophagi. Thousands of years old. They take them, put them in broth, just like the broth you have in there, and they grow. They take them out to space, right? Put them out in the vacuum of space, expose them to the X-rays of space, all the craziness of space, bring them home, put them into broth, and they live. So these are very, very, very resistant structures. In fact, when we think about how to protect ourselves from microbial disease, these are the guys that we're thinking about killing. So when we talk about sterilizing things, when we talk about holding your loop in the flame until it's red hot, that's way hotter than it has to be to kill E. coli. But that's how hot it has to be to melt that spore, allow the heat inside in order to kill the spore. Right? We use an autoclave. We do different things to sterilize. And what we're trying to do is not kill E. coli, not kill that what we call a vegetative cell that's growing. We're trying to kill this guy. And it's really hard. But if we can kill him, we've killed everybody else. So when we talk in chapter 7 about the control of microbial growth, all of those controls, when we talk about sterilizing, are attempts to kill him. Because if I can kill the spore, I will also kill the Mycobacterium tuberculosis, I will kill the E. coli, I will even kill the cell that made the spore. Because they're all less resistant than he is. But sporulation, right, is not for everyone. E. coli can't sporulate, staph can't sporulate, genus Bacillus does. Genus Clostridium, right, which causes tetanus, botulism poisoning, that forms spores. Incidentally, right, it's Clostridium botulinum that you're worried about when you see canned goods, right? You know, moms always tell you, good moms, or you know, moms that are in the know, right? You open up that can of pickles and it doesn't pop, don't eat those pickles, right? You see a bulging can of food, don't eat that food. You know why? Because these guys, Clostridium, actually form spores in response to oxygen. But as soon as the oxygen's gone, they start to grow. And they produce botulinum toxin, which is the most potent natural toxin known to man. And you eat it, and it goes through your intestinal wall, it attaches to your respiratory muscles, and it kills you. Right? Because it's a spore. Because somehow the canning process failed, the high heat, the high pressure of the canning process failed, and you didn't kill the spore. You killed all the growing Clostridium, but you didn't kill the spore. And so once conditions are right, he's like, looks good, and he germinates. And now he's growing again and doing all the nasty things that he would do, and he can make you sick. So all of our sterilization that we do, and we'll talk a lot more about this in chapter 7, is aimed toward killing spores. What do we need to know for chapter 1? That some cells form spores in order to protect themselves from a bad environment. And that could be any number of things. Right? For Clostridium, it's just oxygen, which you and I actually need. So we don't need to know particulars, right? Why do certain cells form a spore? Because they can, and to protect them from an environment that would kill them. All right, a little bit of stuff to wrap up chapter 3. A lot of this is just going to be lab stuff. So pay attention, and I'll try to point out, right? So at the end of today, we will be done with chapter 1, which we finished on Thursday. Chapter 4, which we just finished. And we'll also be finished with chapter 3. What you will notice about chapter 3 is we have looked at very little of chapter 3. Most of what we use chapter 3 for is just to kind of get you ready for lab. Because chapter 3 is all about microscopy and staining, which is what you're doing in lab. So pay attention, right, to the outlines, the quizzes, the online notes to make sure that you're, only focusing on those things that you need to, right? If you don't have a lot, not that any of the stuff we don't talk about isn't important, but right, you want to get a grade, you want to do well on a test, focus on those things. So the first thing is something new that we're going to do with the microscopes today, and that is that we are going to use oil immersion. Woo! Which means you will have to clean your microscopes. And if you don't clean your microscopes, guess who's going to tattle on you? The person who sits in your chair from 8 to 12:30 on Tuesdays and Thursdays. So I will get a little nastygram over here that says there was oil on the microscope. And actually someone gave me a nastygram, and see, I wrote them a nastygram and said, we didn't use oil, and pushed it over to the person who's sitting in your seat before you. But we are using oil today, which means we have to clean up oil today. So why do we use oil, right? So we're using light. We already learned that, right? What allows us to visualize things with a light microscope? Light. That means light, right? So where's the light source on your microscope? It's at the very bottom, right? So the light has to shine through the specimen, into the lens, and into your eye for you to be able to see what's going on. So if you were to take each of those objectives, which you won't do, if you were to unscrew them and look at the bottom, the part that the light goes through, what you would notice is on the 4x objective, right, I put a big red mark there, that's how big the hole is for light to come through. But remember, we learned that as magnification increases, field of view decreases. So look at the difference from 4x to 10x. The field of view is that whole red area, now it got much smaller. That also means less light going into your eye, which is why you move that little slide on the bottom of your stage, right, from 4 to 10. So it increases the amount of light that's coming into your eye to adjust for that change. Then you move to 40x, and now you have a little tiny hole. So you slide that thing over, more light goes in, and what you will probably find with your 40x objective is just the nature of the beast. It's always going to be a little dark and it's always going to be a little blurry, because you are right on the cusp of too little light coming in for you to be able to see. So then we move to the 100x objective, and you have this pinhole. Look at that little tiny red circle. That's how small the hole is to allow the light through. So you could shine like a mag light into the microscope, but so little light would come up through the objective, there wouldn't be enough light to hit your retina, and you can't see anything. Everything looks blurry. So what we do is we add oil, because notice what oil will do, right? So here's the light. Here are all the lenses that are inside the machine. You can't see them, right? This is the one that you can see. And then here's your stage with your glass side, right? Your little knobby thing is right here. Notice when the light comes up, if it hits air, it can refract and go away, right? And that's why when you look, you can see light. All of the light that you can see when you look, like if your view is right here, that's all light that's not being taken up into the objective. So what you can do is add oil, and oil traps the light. Oil traps it and makes it go up into the objective. So notice, and you're going to be using this every time you look at bacteria, you have to look at them with oil. You're going to really want me to come over and look at things. You're going to go, oh, I found it under 10x. And I'm going to say, find it under 100. You can't see them with 10x. 100 times 3 micrometers is 300 micrometers. It's not big enough to see. So we always look at bacteria using that 100x objective, which means we always use oil. And when we use oil, it's going to feel odd if you've never done this before. You are actually going to bring it into focus with your 40x objective. And you're going to look and you're going to go, that objective is so close to the slide. I'm really nervous because she said this is expensive. But this is what you're going to do. You're going to move that objective out of the way, just slide it off to the side, put oil right on your slide, and move your 100x objective right into the oil. You're actually going to have your objective touching oil, which is touching the slide. You will not break your microscope, I promise you, right? That oil objective has a little bit of a spring in it, so it will slide right into place and you can't damage it. What will happen, right? So people will get nervous and they'll pull the stage down and then add oil and then go back up. It's impossible, right? So you're going to bring it under, find it as well as you can with your 40x objective, move the slide just out of the way, right? So you're just going to take it and push it off to the side. So now you have open space, right? So this, you're going to take, and it's going to kind of be at a 45-degree angle. Oil, slide the 100x right into the oil. Do not touch the coarse or the fine focus knobs. All you're going to do is move the objective. Drop the oil, move the objective back, put your eyeballs back, and use only the fine focus, right? And it feels like you're doing something you shouldn't be doing, right? Because it's getting it dirty. That's okay. We're going to clean it. So that is how you're going to view each of your preps today. So you're going to do that 4 different times, right? One with the plate that went to broth, one with the broth that went to broth, one with the broth that went to slant, and slant that went to slant, or something like that, right? Because each time you're going to confirm, do I see only single bacilli? So you get a lot of practice finding little tiny things with the microscope today. One of the things that's going to make them easier to see is that you will stain them. Right? So all cells, those of you who have been in anatomy or physiology know that all cells have a resting membrane potential of -70 millivolts. And then you learn about how cool neurons are, right? Because they can actually change and become positive and blah, 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 blah. And we don't care, but what we do care about is the fact that all cells have a negative charge. And what do we know about negative and positive charges? They're attracted to one another, right? So what do we need to do if we want to stain a cell? Get something that has a positive charge. Whoop, and it gets sucked in, right? It's actually magnetically drawn. Each of the stains that you have are positive stains, which means they have a positive charge, which means they will stain a negatively charged cell. Now, sometimes we want to stain the background instead of the cell. And in that case, we would use a stain that has a negative charge, because a negative charge is going to be repelled by the cell. But all of the techniques that you will use, and I will tell you, and this is really important for lab, not lecture, this confuses people because we're using the term positive here to mean electrical charge. Okay? So a gram-positive cell and a gram-negative cell are both positively stained, because they're both colored. The positive and negative for gram stain means which stain did you hold on to. The positive and negative for an acid-fast stain means which stain did you hold on to. But every time you stain a cell, that is a positive stain. So people will get that confused, right? So for instance, gram-negative cells are pink because they have been stained with the positive stain safranin. And I know, and that's just because for us, positive can mean two things, right? It means the charge of the stain, and it also means the result. It's a negative result in that staining procedure. So positive stains are called positive because they have the colored portion has a positive charge that holds onto the cell. Negative stains have a positive charge, so they stain the outside, right? Or have a negative charge, so they stain the outside. So you, over the next couple of days, are going to be doing some staining in lab, right? Today we're going to do what's called a simple stain. And that's just where you take any stain you want. We're going to use methylene blue and stain cells. It's a positive color, so it will stain all cells blue. We can also, use more than one stain in order to differentiate bacteria. So staining is important because it increases our resolution, right? Remember, resolution is the ability to see 2 points as distinct. It makes it easier to see. That's what it means. It's like putting on glasses, right? That fuzzy E gets clearer when you put on glasses. Staining is like putting glasses on. Staining makes it easier to see bacteria because now they are dark and the background is light, rather than them being light and the background being light. Right? Now, very important, we always have to do what's called fixing bacteria to the slides, because if you don't, you will wash them off. So today's procedure involves heat fixing. That's going to be your most common form of sticking bacteria to a slide, and I'm going to demo that for you today. You heat fix them, it does two things. It adheres them to the slide and it kills them, which means you can take your goggles off because now they're dead. Once you've heat fixed them, you will pour stain onto them, right? And if that is a positive stain, it will be absorbed by the cell and the cell will become distinct, right? It'll stand out from the background. If it's a negative stain, it will be repelled by the cell, and so the background will be dark and the cells will be light, right? But in either case, you increase the contrast between cell and background. We will always use positive stains. There are some techniques for negative staining in your lab manual. We are not going to be using them for various reasons. One, they involve live cells. We can't heat fix them. Two, one of the procedures never works well, and so I don't like making you do something that doesn't work well because that's dumb, right? You can't learn anything from something that doesn't work. So all of the stains we're going to do are positive, right? So you're going to have to keep in mind, how do I know what's positive and negative? It's not whether it has a positive stain, it's whether it holds the first color. Whatever holds the first color is positive, right? So differential staining is like each of these up here, right? This up at the top allows you to differentiate these cells. Now they look different, right? If we used methylene blue, they would all look the same, but now we've used a technique that allows us to say, those are gram negative, these are gram positive, right? This right here is an acid fast stain, which allows us to say these are acid fast, these are non-acid fast, right? Those are important differential stains, right? I said gram staining, most important, right? First identification, gram stain. For antibiotics, for treatments, for diagnosis, we gram stain. Acid-fast stain is very important medically because it allows us to identify whether a patient has tuberculosis. Spore stains, right? So here's a spore stain where green differentiates spores from vegetative cells, which are pink. Spore stains I mentioned, right, because they are the most resistant of all things. They tell us whether or not things are really sterile. And that's mostly important in food preparation, right? So when we sterilize foods, we're looking to make sure that there are no spores. But it's also important for lab safety, right? When you put things in the morgue, we sterilize them to the point where even spores would be destroyed before we get rid of that material, right? Because it would not be safe for us to just, like if after you left, I just dumped it all down the sink. That would not be safe. So we take everything that you use and we sterilize it, and our aim is kill the spores, right? Because that's for safety. We also have a kind of stain. Notice this at the bottom. The capsule stain actually involves positive and negative stains. The cells are stained with a positive stain. The background is stained with a negative stain, and the capsule can't be stained at all. This is the one that you won't get to do because it never works. Right? You can do it perfectly beautifully and it won't work. So you can't really learn much from something that doesn't work for reasons we don't know, right? But it would be very important because capsules allow bacteria to resist your immune system, right? They resist disinfection because of those capsules. So capsules are important indicators of pathogenesis, the ability of a microorganism to cause disease. So this is kind of both lab and lecture, right? So we're going to talk about do each of these labs or these techniques in lab. For lecture, right? What do we use the stain for? To make them more visible. If it has a positive charge, it stains the cell. If it has a negative charge, it stains the background. And we can combine more than one stain to do a differential stain. You will do gram stains on Thursday. You will do, and next Thursday, you'll do both spore staining and acid-fast staining in order to do those two differential stains so that you will learn how to do the techniques that will allow you to identify gram-positive and negative, acid-fast and non-acid-fast, and spores and vegetative cells. Now, one more thing to show you as an overhead, and this is absolutely lab only. This is a light micrograph that I actually took holding over the microscope, which is not, I have a really hard time because some of you guys are great at it, right? But I put E. coli after a simple stain on a microscope and I took that picture to give you an idea of what a simple stain of single bacilli looks like. So when I see this, right, remember I had written over here, I'm looking for three different things. Number one, do I see single bacilli? Yes, I do. Do I see anything other than single bacilli? No. So I have successfully transferred E. coli. I have not transferred any contaminants, so thumbs up to my aseptic technique. So you are going to be evaluating your aseptic technique today, which means we will close the book on lab 2 today when we're done. Because you're going to have to look at all 4 of them first. And I'll come around and we'll look at them, and you can ask other people to look at them too. It's totally fine. You get excited and take pictures and send them to your mom because it's very exciting, right? The other thing that we're going to do is practice, right? I said gram staining is like the most important thing. The other most important thing in all of microbiology is being able to isolate microorganisms, right? Like if you have a patient and there's something going on in their throat and you take a throat swab, the first thing that you want to do, right, you can gram stain it, but you also want to separate all the bacteria that are in their throat, right, because there's normal flora there. And you want to separate all of those so you can see them. And one really good, easy, quick, low media requiring way of separating out bacteria is something called the streak plate technique, which you are going to be doing today. The streak plate technique, so this is just a drawing that I did, right? So notice this is the agar side. The red is where I've labeled. So these are my initials, the date, the medium, which is TSA. I knew that because I looked at the side and there were 2 red lines, and I looked on the back counter and it said 2 red lines mean tryptic soy agar. And then this is a mixed culture. I take a loop and I dip it into the mixed culture. Today you're going to have a mixed culture of E. coli, so you get to see them grow on media, and you've gotten to see them. staph epidermidis. Staph epidermidis is little and white. And E. coli, you might remember from seeing them on the Petri dish, kind of a large colony, it's kind of a beige color. So right now you are going to have, bless you, a mix of both of those organisms. Your goal is to separate them. So you will get a loop full, because job number 1 is to get them from the broth onto the plate. So job number 1 is to take that loop, right? So you'll take your loop, you'll sterilize it, right? You do a cute little pinky thing, wavy thing, dippy thing, wavy thing, pinky thing down. Now your bacteria in the loop. And you go here, and you take that loop and you go over and 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 over, flat, right, flat. So the loop is flat against it, not stabbing like this, but flat like this. Over, over, over. Why? Because my goal is to get as much from the loop onto that portion of the plate as possible. So the blue lines are the lines that my inoculating loop is done. Now, my job is going to be spreading it out. So I don't want any more organism, because that job's done. So now I actually sterilize my loop, and I take my sterile loop, and if I want to, I can stick it right here and cool it. Now I know it's cool, and I take my loop, and I go like this. 1, 2 passes through, 3 passes through. Notice I do not overlap my streaks at all. I do this over, 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 all the way to the end of the plate there, but I don't want to touch the side. If I touch the side, they'll actually grow around the outside of the plate, right? So what I have done is taken some of the bacteria and spread them out. Now I sterilize it again, and I take my sterile loop, and I only go back into the second area, right? So here's my third swipe, is here, here. Notice I'm picking up bacteria. Now I'm spreading them out. All right, I do the same thing. Keep going, never overlapping. Sterilize. And if I have space, I will do this a 4th time. Notice, right? And then really I would do it like this, right? This, over, 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 over. Never overlapping, and very importantly, not going back into that first section. Because what will I do if I go back into the first section? Pick up bacteria. Then I go, I hope it worked. And some of you will be like, I did a terrible plate, can I do another one? I stabbed it, I did all this terrible stuff. No, you can have 1 plate. Because you cannot know how poorly you did or how well you did until you see it grow. It's just like, you're like, I totally screwed up my broth. God knows what's in there. Well, you can't tell until you let it grow, right? We gotta let the bacteria grow. So no matter, and I'm gonna look in the morgue and see, no matter how badly you think you did this, you take that plate and you put it in the incubator. And we will look at them together on Thursday, and guess what? All of you are gonna get to do it again. So please don't think I did a terrible job, I'm gonna throw it away and sneak another plate. You won't learn anything from doing that. What you will learn is streak it, put it in, and we'll evaluate it together. So that's gonna be your job today. So you're gonna do 4 of these simple stains, and you're gonna streak a plate for isolation. Yes?
So we're doing this like this, right? You have to hold it upside down.
So I'm going to show it to you. What I'm going to demo that for you. What I wanted to do is just show you what it looks like, right? And mainly because people will overlap their streaks, so we don't want to overlap streaks. And then the last thing you're going to do today before you go home, right, is clean the oil from your microscope. So today I'm going to give you some supplies. You're going to keep them in your personal boxes. So you've got your lab coats, you've got your inoculating loop. You will be given bibulous paper to blot your slides and make sure they're dry, and you will also be given lens paper. Lens paper, very important, is the only thing that you are allowed to clean the lens of your microscope with. So this is not lens paper. Paper towel is not lens paper. Lens paper is expensive, but it's lint-free, it won't scratch the lenses, right? So you will use this cleaner, right, so that it emulsifies the oil, it breaks the oil up, and then the lens paper will wipe it off. So what we are going to do today to make sure that our microscopes are clean is when you are all done, you're going to leave your microscope on the counter and you're going to say, Krista, can you check my microscope before I go home? That way you and I can make sure it's clean, because this is a learning process, right? So we'll make sure, and if it's not clean, or if you haven't turned the light down or something like that, then I will be able to say, hey, you know, it's dirty right here, right? Let's make that happen better on Tuesday or Thursday. So this will be the last thing you do today, right? So you actually put oil, your lens thinner on that Kim White, and you rub it around the bottom of the objectives. And are you gonna be wearing gloves? No, because you don't need them. You can put them on while you're using the methylene blue today if you have gloves, just to keep your hands from becoming blue. We also have tubes of stuff that looks like a toothpaste tube that's a stain remover that you can use if you get some stain on your fingers, but you are not going to use gloves anywhere near a Bunsen burner. So if you have them and I see you with them near a Bunsen burner, I'm gonna scream and blow a bullhorn at them. I'm not gonna do that, but I'm gonna say take his gloves off, right? Because the most dangerous thing that can happen today is gloves near a Bunsen burner, shrink wrapping to your hands, and then a doctor has to rip it off of your hand and take some of your skin with it when you do that, so don't do that. All right, let's take a break and let's come back at 3:05.